Welcome to the ultimate anatomical transit map. Today we trace how the human body stores, mobilizes, and recycles energy. This is not just a list of random chemical reactions. It is a highly integrated logistical network. It ensures survival under extreme physiological stress. Let's explore how your body keeps the engine running. Your body packs carbohydrates into the liver and muscles, but they handle reserves with opposite philosophies. The liver is the ultimate altruist. It contains glucose 6-phosphatase. This allows glucose to exit the cell and maintain systemic blood sugar. Muscle is the consumer. It selfishly hoards glycogen for its own localized contractions. Muscle lacks glucose 6-phosphatase. Glucose remains trapped inside for glycolysis. Glycogen is a highly branched polymer. At its core sits a protein anchor called glycogenin. It initiates synthesis by attaching the first 7 glucose residues. Straight main chains extend outward via alpha-1, 4 glycosidic linkages. Branch points sprout via alpha-1, 6 linkages. This branching creates countless non-reducing ends. Phosphorylase enzymes can attack from hundreds of angles simultaneously. This rapidly releases massive amounts of glucose during emergencies. When blood glucose is plentiful, glycogenesis stores it away. First, hexokinase or glucokinase traps glucose as glucose 6-phosphate. Second, phosphoglucomutase isomerizes it into glucose 1-phosphate. Third, UDP-GLC pyrophosphorylase activates it into UDP glucose. Finally, the regulatory enzyme glycogen synthase elongates the chain via alpha-1, 4 bonds. Once it hits 11 residues, the branching enzyme snaps off a piece. It reattaches it via an alpha-1, 6 bond to create a new branch. Your metabolism relies on a strict hormonal seesaw. Insulin is the builder. It promotes dephosphorylation. This activates glycogen synthase and inhibits phosphorylase to drive storage. Glucagon and epinephrine are the mobilizers. They activate adenylate cyclase, boosting CAMP and activating protein kinase A. This cascade promotes phosphorylation. It inhibits glycogen synthase and activates glycogen phosphorylase to release energy. When energy is needed, glycogenolysis unpacks the reserves. Step 1 is phosphorylytic cleavage. Glycogen phosphorylase breaks alpha-1, 4 bonds to release glucose 1-phosphate. Step 2 hits a roadblock. Phosphorylase stops 4 residues away from a branch due to steric hindrance. Step 3 is isomerization. Phosphoglucomutase converts G1P back to glucose 6-phosphate. Step 4 diverges. Muscle shoves G6P straight into glycolysis. The liver strips the phosphate to rescue dropping blood sugar. How does the cell clear the branch point roadblock? PP 6-7. It deploys a bifunctional debranching enzyme. First, glucan transferase acts as a crane. It clips the outer 3 residues from the branch and transfers them to an adjacent chain. This exposes the lone alpha-1, 6 residue. Second, 1,6-glycosidase acts as scissors. It hydrolyzes the final alpha-1, 6 bond, releasing 1 molecule of free glucose. Every cleared branch yields exactly 1 free glucose instead of G1P. When glycogen runs dry, gluconeogenesis builds glucose from scratch. It reverses glycolysis by bypassing its 3 irreversible steps. It uses specific enzymes like pyruvate carboxylase, PEPCK, fructose 1, 6-bisphosphatase, and glucose 6-phosphatase. The raw substrates are harvested from around the body: lactate from muscle and red blood cells, glucogenic amino acids from protein breakdown, and glycerol from adipose tissue. This continuous supply line keeps your brain alive. During intense exercise, muscles perform anaerobic glycolysis. This generates 2 ATP for quick movement, but lactate spills into the blood. The liver acts as a metabolic filter. It takes up that lactate, converts it back to pyruvate, and drives gluconeogenesis to recreate glucose. This costs the liver 6 ATP. The net system cost is a loss of 4 ATP. The body willingly accepts this energy deficit to shift the toxic acid burden away from working muscles. During starvation or heavy exertion, muscle proteins degrade. This releases toxic amino groups. To safely manage them, alanine transaminase transfers the amino group to pyruvate, forming alanine. Alanine travels safely through the blood to the liver. In the liver, alanine is transaminated back to pyruvate for gluconeogenesis. The toxic amino group is stripped and pushed into the urea cycle for safe excretion. This preserves local muscle ATP for contractions. Let's contrast these 2 vital recycling systems. The Cori cycle transports lactate. It is triggered by anaerobic conditions and heavy exercise. Its primary purpose is to clear lactic acid to prevent muscle acidosis. It is highly energy negative. The glucose-alanine cycle transports alanine. It is triggered by starvation and protein degradation. Its purpose is to safely transport toxic nitrogen for urea synthesis while salvaging carbon for glucose. What happens when this map breaks? Alcohol metabolism overproduces NADH, forcing pyruvate to become lactate. This blocks gluconeogenesis, causing severe fasting hypoglycemia and lactic acidosis. Von Gierke disease lacks glucose-6-phosphatase. The liver breaks down glycogen but cannot export glucose, bloating the liver. Cori disease involves debranching enzyme failure. McArdle disease lacks muscle phosphorylase, causing muscle cramps. Pompe disease paralyzes muscles via lysosomal defects. Carbohydrate metabolism reveals a beautifully evolved distribution of labor. The brain is the VIP, demanding a constant 120 grams of glucose daily. Every other organ sacrifices to protect this supply line. The liver is the martyr, accepting massive energy deficits and toxic waste processing to keep the body clean. The muscle is the engine, free to selfishly burn energy to help the organism survive. It is a highly evolved network, engineered for ultimate survival.